Good evening, Vanshika. Yes, I'm Rahul Saha, and today we'll be talking about the later part of the chapter. In the last class, we have talked about the various types of androgen receptors, right? So during that discussion, I was telling you that, or I had told you that there are four types of androgens which are present in the male body, right? So we had talked about testosterone. At number two, we had talked about 5 alpha dihydrotestosterone. Then we had talked about androgen. Then we had talked about API. Right, this is your DHT. This is your DHEA. Right. So out of these four types of androgen that we have talked about, I told you that the testosterone and DHT, these are the strong androgens, and they are 95% of them are released from the, you can say, developing graphene follicle, and these two, 95% of these two are released from the zona reticularis of the adrenal glands. Why are they weak androgens? The reason again for that is because there is every time a definitive reason behind every statement in biology. So the reason is, in the cytosol or cytoplasm there is a presence of androgen receptor, right? Just like we had estrogen receptor alpha and beta in case of females. Here we have androgen receptor which is prominent in case of males. So the thing is, in case of the cytosol, the androgen receptor is present in mostly the human males. It is also present in the females, but predominantly it is present in males. And there, the binding of testosterone and 5-alpha DHT to this androgen receptor is very, very prominent or firm or tight binding. Okay, the association constant is high, so they have proper binding, you can say. Thus, they can then be recruited into the nucleus through the nuclear membrane and can act as transcription factors to cause the transcription of certain genes. But the binding of, you can say, DHEA and androstenedione to the androgen receptor is quite loose. This is not tight, okay, not tight binding, okay, or not very, very specific binding. You can call it non-specific in technical sense, non-specific binding. So they cannot elucidate or they cannot go for that dramatic a response or that drastic a response. Okay, good evening, Tarun. That drastic a response when it comes to DHEA and androstenedione. So these two are the weak androgens. Okay, so in PMT or NEET examination, weak androgens are DHEA and androstenedione, and the strong androgens are testosterone and 5-alpha DHT, dihydrotestosterone. Now, I told you that human males, they also have estrogens or female sex hormones in their blood. Don't be surprised. The males have that and the females also have the male hormones. Okay. Now the thing is, for our last discussion, we will be recalling that the blood levels of estradiol, you can say, is perpetuated by what? Is perpetuated by the again developing graphene follicle, but blood level of estradiol or estrogen in human males is very, very, very low. Okay, with relation to the androgen levels. So I had told you that the androgen levels in the last class, if you can correlate. So Let's just talk about the testosterone level, the most predominant androgen and most, you can call it the strongest androgen. So testosterone level was 0.7 microgram per 100 ml of blood plasma. This was discussed in last class. Just to give you a perspective of blood plasma. Now, in case of estrogen in males, it is very, very low. How much low? 0.003 microgram. If I talk about 17 beta estradiol, which is a type of estrogen, it is 0.003 microgram. You can compare 0.7 to 0.003. Okay, per 100 ml. This is in case of males. Okay, in both these levels are in males. So this is the real difference, okay, between the level of testosterone in a boy or the level of estrogen in that same boy, okay, of the same age. Now the testicular sources of estradiol in males are what? A very, very tough neat question. In which part of the testis or which part of the testis is responsible for secretion of, you can say, sorry, which part of the testis is responsible for secretion of estradiol in males? Okay, or testis means males only. So the answer is Sertoli cells. Okay, the answer is Sertoli cells. Okay, write it down. Comparative level question. Sertoli cells, they secrete estradiol in males. Okay, so that is where they will be secreting the estradiol. Now, from biochemistry, we also know that the estrogen, okay, can be converted, or you can get estrogen from testosterone as well. Okay, so some estrogen in males results from, you can say, conversion of testosterone to estrogen in tissues which are other than testis. Okay, like many of the effects of testosterone on bone, okay, it results from the local conversion of testosterone to estradiol. Okay, so just as human females, they can produce both male and female sex hormones, means androgen and estrogens, okay, are also present in, you can say, the blood of males as well. Okay, but the estrogen in human male blood, it do not, or it, you can say, it does not feminize the male. That question you will be asking, why does it not feminize the male? Why don't the male develop the female secondary characters? The answer to that is, I have given the answer in the last class only, that the androgens are present. Okay, just compare the concentration in much, much higher concentration to not let the secondary sexual characteristic, or you can say feature, be expressed through or perpetuated by the estrogen level, because they're very, very low. So that is the stuff that I'm talking about. And estrogen is extremely important for bone growth. Again, so it is required in case of males. You just don't think that it is not required. That's why in tissues other than, you can say, testis, like the bone tissue, we have conversion of testosterone to estradiol. Okay, biochemical pathway in case of bones. Okay, that's why you might have learned, I don't know, that in post-menopausal women, they can suffer or they have high risk of osteoporosis, osteopenia also, okay, in post-menopausal women, because after menopause happens, the blood level of estrogen would do what? Would be plummeting or would get dropped, would become pretty low. And that would then adversely affect the osteoblastogenesis, because osteoblasts, they are produced from the osteoprogenitor cells. They are the bone forming stem cells. And osteoclasts are different. They are produced by the combination of, or you can say, the amalgamation of 50 monocytes. They form one osteoclast. So now they are the bone eating or bone resorptive, you can say bone resorptive cells. So Again, if that balance goes off, you will be losing bone. So first you will be having osteopenia, weaker bone, then porous bone osteoporosis. Okay, that is why in males we do not have osteoporosis as much as women. Okay, because the estrogen conversion rate is almost the same, and we can produce testosterone throughout our life. Okay, that is the and at that same concentration almost. Okay, if we stay healthy. Okay, that is one more reason behind it. Okay, so that is the thing. And testosterone can be converted to estradiol, vice versa. We know that testosterone, if you put aromatase enzyme, just as the aerosol cells after being, you can say, stimulated by FSH, it produces aromatase. So testosterone, if you put an aromatase enzyme, it will convert it into 17 beta estradiol. And testosterone, if you put the enzyme 5 alpha reductase, 5 alpha reductase, reductive mechanism, it will become 5 alpha dihydrotestosterone. That is your 5 alpha DHT, or simply you can call it DHT. Okay, so these two will bind to the androgen receptor, and the 17 beta estradiol will bind to estrogen receptor alpha or beta. Okay, the next topic will be the hormonal control of testicular function. What is the, or how does the testis produce various hormones? Very important topic. Hormonal control of testicular function. So, the male hypothalamus, it produces again the pulsatile secretion, or most of the pulsatile secretion of GnRH, okay, that we all know, which causes the synthesis, and you can say, or the secretion of two gonadotropins, naming FSH and LH, follicular stimulating hormone and luteinizing hormone, from the adenohypophysis. This we all know. But if the anterior pituitary gland adenohypophysis is removed from an adult male, okay, or if the anterior pituitary gland is rendered non-functional because of tissue damage, okay, what happens is the testis undergoes degeneration. No more sperms are produced, and the levels of androgen in the blood drops considerably, because what happens is the Leydig cells, they will cease functioning. Same way, if you get your testis removed, okay, or they become non-functional due to hormonal therapy, that is bodybuilders take. You might have seen the huge body of the Mr. Olympia and all. So let me tell you, what they take is anabolic steroids. The steroids that are biochemically related to testosterone. So that is why, because of higher level of testosterone, one more reason is there, due to higher level of testosterone. Okay. It's a high testosterone level, 0.5 micrograms per milliliter in blood in case of males, especially after puberty, we have bigger muscles. So that is again the discerning point between females and males. You can never see a female, even if she goes for exercise, bodybuilding, even if you watch the Mr. Olympia of the, you can say, women category, you will never be able to see that huge muscles. They will have proper definition: bicep, rectus abdominis, the abs muscles, triceps, quadriceps, everything proper, but the size wouldn't be that huge. Again, the thing is, the males produce more testosterone. That is why we are broader than females. Females are more, you can say, slender, petite in that form. Now, what the males do at that time, also the females can also do, is put testosterone derivatives like diabenolone and all those injections are present. And what happens is, This, you can say, a specific adverse or side effect of hormonal therapies. If you are taking hormone from outside sources, the testis, it does not produce hormone after a certain period of time, because the negative feedback loop, you are completely trimming off or not, you are completely truncating the negative feedback loop, because more testosterone that will go and bind to the GnRH, and then that will not let the FSH be formed. But here you are just injecting yourself with testosterone daily, daily, daily. So testis stimulation, non-stimulation, you are just taking it out of the equation. So what will happen is testis undergo atrophy. Their testes become smaller in size, and ultimately they cannot make testosterone. So in order to make testosterone, they are completely dependent on hormonal injections throughout their life. That is the story of bodybuilders, right? But if a normal person will take out their testis, okay, what will happen is the androgen levels will be very, very low. Okay. Now, restoration of full fertility and androgen production of a male where adenohypophysis has been taken out, it will require administration of either both FSH or LH from outside sources, or you can say he will not be fertilized, or he will not be able to become fertile, because even if you put one FSH or LH, that will not do the work. You have to go for both. Okay. So the thing is, in humans and other mammals, what happens is LH, it stimulates the Leydig cells to secrete testosterone. Again, one great neat question. Okay, LH stimulates luteinizing hormone Leydig cells to secrete testosterone. Okay, that is the work of especially the luteinizing hormone in case of the males. But this androgen does not enter, okay, it the blood circulation directly, okay, it enters the blood circulation through, you can say, androgen binding protein, and goes to other areas of the body, and also it diffuses through the testicular interstitial spaces through the basement membrane into the seminiferous tubules that I was talking about in the last class. That through these are the interstitial cells only, the Leydig cells, and through that they'll be diffusing, and will be diffusing through the basal membrane, then reaching the epithelial cells, specialized epithelial cells known as the seminiferous tubules. And testosterone levels in the testis thus are about 100 times higher than they are in the general circulation. Okay, so much high testosterone level is found in the testis itself. Okay, now why is it so high? Because that much high concentration of testosterone is required for the process of spermatogenesis. But again, the male germ cells, they lack androgen receptors. Okay, the male germ cells, same way like I told you for the follicular phase. Okay, how does they, you can say, how do they know the GnRH neurons? Okay, the specific level of FSH. So I told you that the GnRH neurons, they do not have FSH receptors. They are present in the nearby cells. Okay, but they have the receptors for the kisspeptin. Same way here, the male androgens, or sorry, the male germ cells, they also lack androgen receptors. A great, great theory. The male germ cells, they lack androgen receptors. The same way as they lacked, or the GnRH neurons, they never had the receptor for, you can say, estrogen. Okay, that they will be able to know the level of estrogen and all. So they also lack the androgen receptor. Now, when they lack androgen receptor, what would happen? The testosterone cannot affect spermatogenesis directly or not. The testosterone cannot affect spermatogenesis directly. Okay. Now this is again the case. Now what is the detour? Sertoli cells, they possess androgen receptors. Now Sertoli cells, they possess the androgen receptor. Very, very important, okay, for competitive level. Sertoli cells, they do possess androgen receptor. Now, receptors for FSH are also present on these Sertoli cells. Also, FSH receptor. Both the receptors are present on the Sertoli cells. And what does FSH do? It appears to amplify, you can say, the spermatogenesis promoting effect, okay, of the testosterone hormone. Now, testosterone together with FSH, what it does is it supports the survival of the spermatogonia as well as spermatocytes by suppressing the cell death of the germ cells. They suppress the cell death of the germ cells or the male germ cells. Cell death is known as apoptosis. Now, one more thing is prevention of cell death may occur. Why? Because these hormones help maintain the intimate associations between the developing male germ cells and their Sertoli cell support system, that they are completely associated, that is also maintained in the process. Now, testosterone along with FSH, that is again a postulate, okay, it supports what? It supports the meiotic progression of the germ cells and allows Sertoli cells to sustain contact with spermatids as they differentiate into, you can say, spermatozoa. That is one more theory. Testosterone along with FSH, you can say possibly along with FSH, they induce meiotic division. Meiotic division in the germ cells and there, and also you can say it maintains the, you can say, contact of Sertoli cells with spermatids. Maintain the contact of Sertoli cells with spermatids. That is one more theory, and that will lead to, you can say, differentiation of spermatids into spermatozoa. So that will lead to spermatids differentiation into spermatozoa plural or spermatozoon singular. Okay. Now, what else is done? Testosterone also promotes the release of mature sperm during spermiation, and also FSH helps in this process. Now, you can say these are the functions of testosterone, and many functions are also which are not in your course. I told you that the testosterone can be converted to estrogen also, right? By aromatase. So that is also happens. So testicular estrogen is also produced, okay, by aromatization or by the action of aromatase enzyme in the testicular cells. And they can also play some important role in spermatogenesis, okay. Now, in the or under the influence of testosterone and FSH, the Sertoli cells, okay, they secrete androgen binding protein. They have the receptor for both. Now, under the influence of, you can say, testosterone and FSH, the Sertoli cells secrete ABP, androgen binding protein. That is your ABP. And this protein does what? This protein is secreted directly into the fluid of the tubular lumen, where it binds to testosterone also and DHT also, like dihydrotestosterone 5 alpha. So directly it is secreted into the tubular lumen of the seminiferous tubular lumen, and will bind to both testosterone and 5 alpha dihydrotestosterone DHT. Now the role of ABP is what? Is to concentrate androgens near the Sertoli cells. Okay, near the Sertoli cells, so that these steroid hormones can, you can influence spermatogenesis more effectively. And you can say furthermore, these androgens are kept from diffusing out of the tubules. Okay, why? Because the blood testis barrier is there, I told you, which is again perpetuated by or maintained by the, you can say, Sertoli cells only. And the blood testis barrier, it would hinder the passage of ABP, androgen binding complex. So just think of the evolutionary pathway here, that testosterone and FSH, mostly this testosterone, can diffuse out of the blood testis barrier. Blood testis barrier. If it does diffuse out, it will not be able to bind with Sertoli cell for a long period of time. But it can diffuse out. But when testosterone and dihydrotestosterone 5 alpha dihydrotestosterone DHT, they bind with, you can say, androgen binding protein, then the size becomes huge, especially of the androgen binding protein, and then they cannot diffuse out of the blood testis barrier. Think of the evolutionary significance of this, meaning that they will stay concentrated in the, you can say, or towards the other side of the Sertoli cells, and will act with the concomitant, you can say, participation of Sertoli cells and the male germ cells. So that is how they will stay concentrated in the Sertoli cells. Okay, so that is the entire zest of this specific discussion. Voice is lagging. Here it's come. Wait, let me just check. Just let me check. No, here my settings are fine, Amita. Completely fine. Okay. Just check your setting once because my settings are completely fine. And don't worry, from next week onwards, we'll be having the offline classes. So this technical issues is that they sometimes creep in. They will automatically be resolved, right? So next, this the control of gonadotrophin secretion in the male. Next topic, control of gonadotropin secretion in males. Okay, okay. In males. Okay. So the blood levels of FSH and LH in male, they remain relatively constant. Okay, there is not like that they will be too much, you can say, differing. You can say FSH and LH, so they'll remain relatively constant. There is nothing like LH surge which will take place, right? That will, the excessive, you can say, estrogen will specifically stimulate the anti-reproduktory gland to secrete LH only, which happens in case of women. So that does not happen in male. But there is some evidence that levels of LH and testosterone, they exhibit some kind of daily cycle. Okay, like being almost 20% higher in the morning. Okay, so we already have seen the feedback loop, negative feedback loop of the steroid hormones that regulate FSH and LH in females. Okay, now if testosterone is given to an adult man, okay, LH levels will automatically go down and FSH levels are also reduced to a lesser extent. Okay, so you can say that testosterone exerts a negative feedback at least on LH secretion. Okay, they exert a negative feedback on LH secretion. So GnRH from the hypothalamus, okay, then pituitary gland, here FSH, LH both, and then it will go to, you can say, testis, Leydig cells, then testosterone. Now, if you are given testosterone from outside, means excessive testosterone in case of bodybuilders, okay, that will go to the anterior pituitary gland and specifically suppress, you can say, LH secretion. Okay, so this is one more, you can say, comparative level question, that it will suppress LH secretion. And under normal conditions, what happens? When testosterone levels drop, LH levels rise, and they will stimulate whom? They will stimulate the specific cells that I have written down here, the Leydig cells. Okay, and the Leydig cells will then again cause more and more testosterone secretion. And the opposite of this is happening in this negative feedback loop. Okay, when testosterone levels rise. But when excess androgens are administered in adult men, they can become temporarily infertile. Okay, that sperm synthesis may not be continued, but their, you can say, sexual drive is not reduced. But if this happens continuously, okay, then complete, you can say, infertility can, you can say, set in case of, you can say, the pro level or the professional bodybuilders. Now testosterone, it regulates LH secretion by influencing what? By influencing GnRH release, okay, from the hypothalamus, and by affecting the sensitivity of LH secreting cells again in the anterior pituitary gland to GnRH. Both these things it can control. Now, the question arises is the LH negative feedback is still clear, but what kind of feedback systems are present that regulate the FSH secretion in male? Okay, what kind of negative feedback loops are present that regulate the FSH secretion in male? Now, here the evidence is very unclear or less clear, you can say. But it appears that FSH is controlled by two negative feedback systems that I can think of. First is, you can say, high testosterone level will exert negative feedback on FSH secretion also at the hypothalamic and pituitary levels. And again, in addition, if you secrete high testosterone, the Sertoli cells will secrete inhibin. This is a glycoprotein which is secreted in response to very high FSH. Now again, that will play a huge role in FSH release. It will decrease the FSH release. Now I told you in the case of women as well that inhibin suppresses FSH but not LH secretion. Okay, like possibly by, you can say, it can decrease the sensitivity to GnRH of FSH secreting cells in the anterior pituitary gland. Right, this was already told. So quite on the same track, FSH feedback, okay, in males. How is FSH regulated? So here, hypothalamus, then GnRH, then anterior pituitary, here FSH, and LH, and then testis, testosterone. Okay. So when there is too much FSH, okay, it will automatically go and bind here. Okay, and what will happen is too much FSH secretion, okay, directly will have the hypothalamus and pituitary will bind here and here both, okay, and also the Sertoli cells, okay, in the testis. You have the Sertoli cells, they can release inhibin. Inhibin selectively goes and binds to the anterior pituitary gland and only downgrades, you can say, or reduces the level of FSH, okay, not LH. So that is again one theory in case of males, okay. Now there is also some evidence that activin, which I have told you, which will increase the effect of FSH, again a protein released by Sertoli cells only, will stimulate the effect of FSH as well as its local effect on the, you can say, testicular cells. Okay. Now this is the complete picture of the you can say hormonal regulation in case of males. Okay, now the thing is, one more thing I'll tell you is, why do you need scrotum, or why does a male need scrotum? Okay, number one is the temperature hypothesis. Like, you can just think of the temperature within the scrotum is 3.1 degrees Celsius to be exact, lower than normal internal body temperature. Now, this hypothesis is, you can say, tells us that the testes are external because spermatogenesis cannot occur at the core body temperature. Okay, so this is what's written in your book. But the thing is, if you check evolution, okay, so the ancestors of mammals with external testes presumably had internal testes as well. So I can say, why didn't scrotal mammals retain the testicular enzymes that function at high temperature and keep the testes inside? Because they had internal testes also. So then this theory cannot explain testicular descent in mammals with internal testes. Why couldn't they, as per evolution, start retaining the testes inside? Because we have achieved such so many enthralling things via evolution. Why not this one? Next, I can talk about sperm fitness also. Now, this one tells us that sperms are produced and stored in the epididymis and vasa differentia in cool scrotal temperatures, because this environment would improve sperm quality. Now, the cooler temperatures, they induce the formation of more mitochondria in each sperm midpiece. With these mitochondria, you can say, providing more energy to the sperm tail beat, when the sperm would require to move up the female reproductive tract. Okay, so that is one more theory that I can think of, right? That the sperm are in the cool scrotum. Okay, temperature of scrotum. Okay, the 3.1 degree lower than the body temperature or core body temperature. So that would lead to more mitochondria production, synthesis in the mid piece. The mid piece. Now that would lead to, you can say, proper cellular movement from tail. Okay. Now, one thing is this one. Now, evidence for this is the fact that in mammals with pelvic internal testes, okay, the epididymis and vasa differentia with their stored sperm, they, you can say, project toward the body wall to a scrotum like skin out pocketing. But you can say, however, mammals with abdominal internal testes, okay, seem to just do fine without this specific sperm cooling mechanism. Again, why can't they become infertile, or why haven't they become infertile, and why do we require scrotum? Because the internal testes possessing mammals, they are able to produce fit sperms. So you cannot say that even the sperm or the fitness of the sperm is directly related, okay, to the scrotum being descended, okay, in case of any male. So that is again a, you can say, great hypothesis, but again this lacks. Okay, so I have no answer, okay, personally, The books have written these two answers, but I have the counteract of that. Okay, that I told you. There is, if you think deep, why evolution, then still the scientists are thinking of why does the scrotum hang down. Okay, via evolutionary perspective. Okay. Now, what does deficiency of nutrients do, or how do they take place, or what kind of, you can say, ramifications or consequences that they induce? So the specific nutrient composition in the diet, it may also affect the testicular function. Okay, a severe deficiency of certain micronutrients like vitamin A, D, E, and zinc has been shown to decrease male fertility. Okay, so specifically the males, they need to take these vitamins regularly through their diets. So zinc deficiency leads to male infertility. Okay. Now, women who exercise intensely can also become temporarily infertile. Okay. Now, the effects of exercise on men are less clear, you can say. But again, exposure to toxic substances or certain types of radiation can also adversely affect the testes, can cause spermatogenesis to be ceased or even direct infertility. Like certain chemicals are there which can damage the testes completely. Like for examples, we have heavy metals like mercury, lead, manganese. Okay. So these are also found in some Maggie and all, right? Heavy metals. They were found and then Maggie came back into action again into the market without proper, you can say, scrutiny. So certain heavy metals like mercury, lead, you can say manganese, cadmium, okay, they can block blood flow to the testis. They can directly block blood flow to the testis and then will cause sterility, infertile, okay, irreversible sterility, you can say. And same way, the environmental pollutants like organophosphate based, you can say, pesticides can also have adverse effects on testicular function. Then if you have seen or watched the Avengers and all, so high levels of radioactivity and chemotherapy. If you are going for testicular cancer treatment, then you will be taking chemotherapeutic drugs. Okay, it can also destroy the type A spermatogonia. Okay, so the chemotherapeutic drugs can destroy the stem cells, right? The unipotent stem cells will only become type B spermatogonia, not any other organ. They will not be the unipotent stem cells here. Okay, so known as type, can destroy type A spermatogonia. Thereby, you cannot produce sperms for the rest of your life. Okay. Now, exposure to X-rays can directly destroy the germ cells. Okay, sometimes leading to temporary reduction in fertility. If you are getting X-rays done regarding in your thigh area, or you can say the pelvic bone injuries repeatedly, that can lead to temporary reduction in the fertility. Again, certain viruses also can infect the testis. Okay, like mumps virus, which causes painful inflammation and enlargement of the, you can say, salivary glands. This virus has also been seen to affect the post-pubertal male's testis. Okay, even the SARS-CoV-2, you might have seen some research papers and some news also, that this can also invade testis, the blood testis barrier, and can also cause infertility. Even the SARS-CoV-2, okay, that causes COVID-19, can cause infertility in males. There are multiple research papers and also multiple reports of this happening in males, that they can cross the blood testis barrier and it will be able to directly, you can say, cause sterility in males. Now, we have seen the use of anabolic steroids that I was talking about in today's class only. So the widely available anabolic steroids are androgens and its derivatives. So they are taken by men and sometimes also women who are intent on building their body in high amounts to increase muscle size and strength. Okay. Anabolic means they'll go for anabolism, means muscle synthesis, muscle mass buildup. Anabolism and catabolism, because it's two ways metabolism works in our body via biochemistry. So anabolism is synthesis. These are the reductive processes. Catabolism, like glycolysis, is, you can say, breakdown or oxidative processes. So anabolic steroid, because these hormones are steroids in nature. So you can call them the derivatives of androstenedione. Okay, these are taken by males, sometimes also females. Okay, in high amounts that I was talking about to increase muscle mass. Okay, to increase muscle mass, and the thing is, these drugs do increase muscle mass, but or unfortunately they can have very serious side effects. Okay, in males, this excess androgen in the system, I told you, it will reduce luteinizing hormone secretion from the pituitary gland, which will lead to testis, testis shrinkage and infertility. So one question is asked that it directly reduces LH secretion, no Leydig stimulation, ultimately testis shrinkage, okay, and infertility. This happens in males. Okay. Also, if you keep on taking them in, you can say, for a chronic period of time, you can have acne, you can have liver tumor, you can have kidney disease. Okay, you can even have high blood pressure, hypertension. Okay, then even cardiomegaly, increase in the heart size, hypertrophy of the, you can say, muscles, increase in the heart size, that will lead to cardiac failure. Okay, congestive heart failure. You can have hypertrophy of heart. Okay, that's why most of the bodybuilders, they die at the age of 40 50. Those are the intense bodybuilders. They'll go to their washrooms and they'll be found dead. Okay, and then after the post-mortem, after the examination, you will see their heart is that huge. Okay, like really huge, that it shouldn't be. It should be as the size of your right fist mostly. But they have really huge heart. Okay, even sometimes they take growth hormones along with this, so that is more, you can say, destructive. It will increase the size of your testes because you're taking growth hormones, so they increase the foot size. Their, you can say, their shoe size is increased, their head size is increased, even their large intestine is, it it grows, their small intestine grows. You can say lots of difficulties they face. And for women, they again can become infertile and suffer the same effects, plus beard growth, voice deepening, because testosterone level is very low in women and you're increasing, so they'll become kind of masculine. Okay, reduction in breast size, that is known as breast atrophy. Okay, so those things can happen in women. Okay, and then aggression issues in men and women both. Okay, depression issues also, psychosis also, these things are, you can say, prevalent if you keep on indulging in these kinds of supplements of anabolic steroids. So I hope this lecture was clear to you. Now, only the cancer, testicular cancer, and you can say the entire structure of the male accessory ducts and all that is remaining, so that we will cover up in the next class and finish off this chapter. Okay, then. Thanks a lot. Let's